Okay, hi everybody. So this is the DNA replication lecture from the Medical Genetics module, Level 3 module, which forms part of the Biomedical Science degree at the University of Bradford in the UK. So what we're going to be talking about today is, I guess, a whistle-top tour of DNA replication in eukaryotic cells, so in human cells, if you like. And the overview of the lecture is thus. We're going to start with just a brief introduction to DNA replication and some general reminders about why, obviously, it's such an important process. We're going to consider the replication checkpoints. We're going to cover in very brief detail a couple of things about the cell cycle control behind the cell's decision whether or not to replicate its DNA, why that's important. Before moving on to some basic chemistry of DNA replication. I'm no chemist, so I'm not going to get into vast amounts of detail, but it's important you understand some of the chemistry behind how DNA replicates. And then we're going to talk in a lot more detail about the DNA polymerases and the replication fork structures, which are obviously the proteins that are responsible for the actual mechanical act of replicating DNA. Once we've talked about how the nuts and bolts of how DNA is replicated, we'll consider how replication's controlled, and by that I mean how the cell controls how many times DNA is replicated during a cell cycle. In other words, how the cell controls the fact that DNA is only ever replicated a single time during a cell cycle, how that works, before finishing up with a consideration about the so-called End Replication Problem. So this is an issue that is a particular problem when copying or replicating linear pieces of DNA, such as eukaryotic chromosomes, and how telomeric structures and telomerase are involved in circumventing this end replication problem. And to finish with, we'll discuss a couple of examples of human diseases which arise due to mutations in genes which are responsible for the DNA replication machinery, either directly or diseases which arise due to issues and errors with the DNA replication process itself. So the learning outcomes of the lecture then are, you should be able to explain how a cell regulates the initiation of replication. So in other words, you should have a broad understanding of the cell cycle control mechanisms which exist, which either commit a cell to replication or prevent it from entering S phase. You need to understand the underlying chemistry of DNA replication and be able to discuss how DNA polymerases and other proteins function during replication to achieve DNA replication and to duplicate chromosomal DNA. You need to explain what the origins of replication are, why they're important, and importantly how they function to ensure that DNA replication is tightly controlled during the cell cycle. And you should be able to also explain what the end replication problem is and how telomerase and telomeric DNA sequences overcome that problem. You also need to be able to give some examples of diseases and human diseases in particular, obviously, which arise due to errors in the DNA replication machinery or process. So DNA replication. Well, obviously by now I would hope that most of you understand that DNA is the molecule that harbours obviously the genetic code. So the traits which are passed on through generations, the information for those traits obviously is contained within DNA. It wasn't always so obvious that that was the case. Obviously, going back just 50 or 60 years, it was quite a contentious idea that DNA was able to achieve such an impressive or fulfill such a complex, impressive role within the cell, because it's a very simplistic molecule essentially. And it wasn't until scientists realised really that DNA is self-complementary that the penny kind of dropped with regards to the fact that here was a molecule which was really, in a very simple, elegant sort of way, going to be able to pass on information and replicate that information through generations of cell division. And so it's this concept that you have two complementary strands of DNA that are base paired together in a predictable way. In other words, A's and T's are always found opposite one another, and C's and G's are always found opposite one another. And the concept, therefore, that if you were to split these two strands apart, that each strand itself could act as a template for the generation of a complementary copy of that strand. So that's the basic premise of DNA replication. Obviously, it's a really obvious, elegant system. It still had to be proven, and it was proved by a number of real seminal pieces of work in the 1950s and 60s, one of which you cover at level 1, the Michelson and Stahl experiment. And it's these sorts of classical molecular biology experiments that really paved the way for us to understand how DNA was truly able to be the molecule which contained the genetic information which is passed on through generations. So in principle, the replication of DNA itself is very straightforward then. The strand acts as a template for the production of a new strand. We've talked about PCR on the molecular genetics module last year, and the fact that PCR, one of the beauties of it is that it's a very simplistic reaction to run in the test tube. Here's a reaction where to make new DNA, all you need is a template, a DNA polymerase, and the free deoxynucleotides which are incorporated into the newly synthesized strand of DNA. So in an in vitro situation in the lab, it's a very simplistic process to actually recapitulate. In practice, however, when you start considering the replication, the synthesis of entire chromosomes in particular, but also smaller genomes such as those which occur in bacteria and viruses, the process is actually incredibly complex. What I've got here is an scanned electron micrograph of DNA actually undergoing replication. This is a double-stranded DNA helix here. As you can see, it splits into kind of two tails, and what this represents here is a replication fork. So it's at this kind of juncture, this Y-shaped juncture, where all the action's going on with regards to DNA replication. And it's this replication fork structure that we're going to talk about in a lot more detail later on in the lecture. So before we get into the mechanics of DNA replication, it's important to consider how cells decide whether or not to replicate their DNA in the first place. It's not melodramatic to state that the actual passing on of the genetic information is probably the most important process a cell will decide upon or will perform. Yeah? So the concept of the selfish gene obviously is something that you've probably encountered. This concept that organisms exist to pass on their genetic information to their offspring, and that's a driving force of evolution. Similarly, cells want to divide, want to produce daughter cells, but all these processes need to be tightly regulated. Yeah, there's no point dividing at a time which is going to result in catastrophic events occurring within the cell and cell death. Similarly, I'm sure that many of you will want to think very carefully and ensure many checkpoints are in place before you decide to pass on your DNA and replicate, so to speak. So obviously it's an incredibly important process, one that requires tight regulation. For example, in simple eukaryotes such as yeast, which is the simplest eukaryotic cell really, the decision whether or not to divide, whether or not a cell wishes to replicate its DNA and then undergo a full round of cell division is primarily controlled by the availability of nutrients. So it's a very straightforward situation. If there are yeast cells growing in the lab on an agar plate and there's plenty of nutrients available in the agar, the yeast will happily divide. So in other words, cells will undergo constant cell cycle division whereby the DNA is replicated, cells undergo mitosis, get two new daughter cells produced which grow, will replicate their DNA, undergo mitosis, and so on and so forth. The situation in our cells, in eukaryotic cells and mammalian cells, is a lot more complex. It's more difficult than just a simple determination of available nutrients. There are many other factors to take into consideration, particularly in multicellular organisms. And in fact, the signals which determine whether or not our cells divide are often extracellular signals, which are mediated via intracellular signalling mechanisms, which you may have encountered already on the course, or you'll certainly touch upon next semester in biology of disease. And it's these extracellular growth factor signals, really, which often are the driving force for whether or not a cell will divide and enter the cell cycle and undergo DNA replication. How is all this process regulated? If you lift the hood on this, if you lift the bonnet up, what's the mechanics that are actually controlling this decision of whether the cell is going to decide to enter the cell cycle, go through G1, and then critically pass its restriction point here in G1, at which point the cell is completely committed to the replication of its DNA and separation into two daughter cells? How is that controlled? Well, before we get into that, there's just a classic experiment I want to make you aware of that really pointed scientists in the right direction with regards to all of this. And this was some work done in the 70s by Johnson and Rayo. And what this work suggested, or what the data from this work suggested, are two things. It suggested that factors exist in the cell that can drive cells into S phase. So things exist in cells which are able to drive cells forward through the cell cycle. At this point, it wasn't clear what these factors were, but these experiments prove that they probably exist. The second thing that these data suggest is that these factors which drive the cells forward into S phase, well, there are also additional factors that must exist which make sure that DNA only replicates once per cell cycle. So how did they come to those two conclusions from these simplistic data here? Well, what they did is they fused cells together which were known to be at different points within the cell cycle. And they asked the question of what happened to the individual cells once these cell fusions had occurred. So by fusing these two cells together, what you do is you mix the cytoplasmic contents of these cells, you mix any factors which might be present in the cell here with factors which are present in this cell here, and you ask the question of whether or not cells in different stages of the cell cycle can influence cells which they're fused with. So here we're fusing a cell that's in S phase with a cell that's in G1. So this is an S phase nucleus. It's getting ready. It's actually in the process of replicating its DNA. This is a G1 phase nucleus here. So this hasn't started replicating its DNA yet. In fact, it may not have passed and probably hasn't passed this restriction point. However, when you fuse the two together, what you see is very rapidly this G1 phase nucleus is driven into S phase. Yeah, even in the absence of external nutrients or anything else, the fact it's been fused with this S phase cell drives a G1 nucleus into S phase. So that tells us that there must be factors present in an S phase cell which are capable of driving cells from earlier on in the cell cycle into the S phase of the cell cycle. Similarly, or in contrast I should say, if you look at a S phase cell here and you fuse that to a G2 phase cell, so this is a cell that's already replicated its DNA, it's not divided yet, it's not undergoing mitosis, but the DNA content of the cell has been replicated. Yeah. Now we see something very different. The S phase cell here isn't capable of driving this G2 phase nucleus back into S phase. The nucleus stays in this G2 state. Yeah. So that's what that tells you is whatever factors are present in an S phase cell which can drive a G1 phase cell into DNA synthesis, those same factors cannot drive a G2 phase cell into DNA synthesis. Or to say that in another way, once cells have replicated their DNA once and they're in G2, there is something there that prevents that process from happening again. Yeah, and for good reason. You don't want to continually replicate your DNA. That's how you end up with aneuploidy, abnormal numbers of chromosomes. You only want to replicate them once per cell cycle. And the cells obviously have some way of making sure that that's the case. Even in the presence of all the factors that normally drive cells into replicating DNA, if you take a G2 nucleus which has already replicated its DNA, you can't artificially drive that back into the S phase again. It cannot happen. There's a break there that's applied. So cell cycle control is a complicated process in its own right. We touched upon this at level 1 in human genetics. I'm not going to go into much more detail of the rest of the cell cycle control in this lecture. It's beyond the scope of this lecture. But what I do want to talk about is S phase entry in a little bit of detail, because it's obviously directly relevant to what we're talking about today. So a major player in the control of entry into S phase, which the more conscientious of you might remember from human genetics, is this retinoblastoma protein, or the RB protein. So it's a master regulator of S phase entry in eukaryotic cells, in human cells. And what it does is actually, in G1 phase cells, RB works in a negative fashion to prevent entry into S phase. So RB controls S phase entry by effectively inhibiting it in G1 phase cells. And it inhibits it by interacting with this other protein E2F, which is a transcription factor. Now E2F is a very important transcription factor. It's a transcription factor which is responsible for binding to and switching on expression of a whole range of genes which are required for DNA synthesis and S phase entry. So if RB is bound to E2F, this E2F transcription factor isn't capable of turning on gene expression for all these key DNA replication proteins, and therefore DNA replication cannot proceed, S phase entry cannot occur. However, in response to external factors in our cells such as growth factors and intracellular signalling pathways such as the RAS signalling pathway, what eventually can happen is you end up getting activation and production of a certain set of CDK cyclin-dependent kinases. And these cyclin-dependent kinases function to phosphorylate the RB protein, and by phosphorylating the RB protein you perturb, you disrupt the RB protein's ability to interact with the E2F transcription factor, thereby releasing this transcription factor to turn on gene expression of all these S phase entry proteins and enzymes that are required for DNA replication. So in other words, once you get a growth factor sending a signal, cyclin-dependent kinases phosphorylate RB is released from the transcription factor, and the transcription factor then goes about the job of turning on all the genes which are required for S phase entry. So it's quite a straightforward, simple process, and it's a major mechanism for controlling S phase entry into eukaryotic cells. It's a little bit more complex than that, it always is. And another thing I just want to mention with regards to S phase control is the fact that RB doesn't operate in a vacuum. It doesn't operate in a nice simple pathway like I just showed. It operates within a complex network, a complex feedback mechanism, which also involves, among other things, two other genes, P53 and CDKN2A. So P53 you will have heard of, CDKN2A you may not have heard of. It's an interesting gene that actually produces not one but two transcripts, P16 and P14, which along with P53 you can consider and think of as DNA damage sensors. So you should know by now that P53 is it's colloquially known as the guardian of the genome. It's responsible for monitoring DNA damage events. It's one of its jobs. So damage to the DNA. When P53 detects a DNA damage event, one of the things that it does is it increases the production of this protein P21, which in turn is able to inhibit the cyclin-dependent kinase which normally phosphorylates RB. So as we listened to on the last slide, RB phosphorylation is required in order to disrupt RB's interaction with E2F and allow the E2F transcription factor to turn on the genes required for S phase entry. So if you inhibit the thing that normally phosphorylates RB, you are in fact inhibiting entry into S phase. And that's what P53 and P16 and P14 actually do. They, via different mechanisms, eventually lead to the inhibition of S phase entry. The reason P53 wants to do that is it wants to make sure that any damage to the DNA of a cell is repaired before the cell decides to replicate the DNA. The worst thing that can happen to a cell and ultimately to the organism from which the cell belongs is that the cell replicates damaged DNA, because that's how you effectively establish and pass on mutations, which can ultimately lead to disease. So P53 is very important because it prevents the incorrect replication of DNA in cells which have undergone DNA damage. And in fact, loss of, as you will also know, loss of the P53 protein is one of the first events which occur on the road to cells becoming cancerous and cancer developing in patients. So it's a very important mechanism, S phase entry control. I don't want to talk any more detail than that about it today. Okay, so I said earlier that it's important you understand a little bit about the chemistry behind DNA replication, and we're not going to go into a lot of detail because I don't understand the chemistry that well myself, but it's important you understand some of the chemistry of the process, because it kind of informs with regards to why the process works in such a way that it does with regards to the replication of the DNA. The thing I'm skating around the topic a little bit. What I'm trying to get at is one of the key tenets of DNA replication is the fact that the DNA can only be replicated, can only be synthesized in a 5' to 3' manner. So let's deal with this first before we get onto this formula. So this has been told to you before when we've talked about PCR, the fact that DNA synthesis always occurs in a 5' to 3' manner. The reason it occurs in that direction is down to the chemistry involved in the actual addition of nucleotides to a growing single-stranded piece of DNA. And this addition works in essence by the fact that the triphosphate region of the incoming trinucleotide phosphate, the DNA base, is incorporated into the single-stranded growing DNA template by virtue of these triphosphate groups here on the 3' end of these 5' triphosphate groups here on the incoming nucleotide attacking this 3' OH group on the growing nucleotide strand, on the growing DNA strand. So these 5' triphosphates attack the OH group here on the DNA strand, and that causes the formation of a phosphodiester bond between the incoming nucleotide and the existing DNA molecule, and the release of pyrophosphate, two of the phosphate groups, from this trinucleotide phosphate base. So what you get is incorporation of the base here and release of pyrophosphate here, which are two of these 3 phosphate groups. So to get onto this chemical formula here, or this chemistry formula here, which kind of just gives a formula interpretation of this cartoon, this is the growing nucleotide. This is the nucleotide strand, or the single-stranded piece of DNA that's been increased in length, that's been synthesized. This is what this represents. These are monophosphate bases, one phosphate group, X monophosphate here, and N obviously refers to the length of the chain. This is the incoming triphosphate here. So what you do is you add the incoming triphosphate to your existing DNA strand, and what that results in is this strand is extended by 1. So you have XNP plus 1, N plus 1. And this 2-phosphate here is the byproduct of the reaction. So once pyrophosphate is cleaved, you're left with 2 phosphate groups which can be recycled. Now the conversion of pyrophosphate is really the thing that I want to emphasise and hammer home to you. This is the key event, really, if you like, kept from a chemical point of view, with regards to the synthesis of DNA. And it's important because it's this event which actually creates an energetically favourable situation during DNA replication. So what I mean by that is if you remove the actual conversion of pyrophosphate into 2 phosphate groups, the energy, net energy of this reaction is pretty poor. Now when you've got a reaction, a chemical reaction, a biochemical reaction which has got poor net energy, a low K value, it means that the reaction can tend to seesaw. It can tend to be reversible and work both ways. Now obviously if you're a cell trying to replicate all your DNA and you've got a lot of bases to replicate, the last thing you want is to replicate, say, 100 bases and then suddenly 30 of them get unreplicated because the reaction reverses and those bases start falling off. So to ensure that that doesn't happen, the reason why that doesn't happen is that in addition to the actual addition of this base here to the growing chain, you also get cleavage of this pyrophosphate molecule, which is highly energetically favourable and lends an overall high K value to the entire process of adding nucleotides to growing DNA molecules. So what that means is the energy is such that never will you see DNA replication working in reverse. Once you've replicated a nucleotide, it stays replicated because the K value of the process is very high. Yeah. So that's really, I don't want to get into more detail than that with regards to the chemistry. One other thing then I want you to start thinking about though is how the cell makes sure that DNA replication proceeds correctly. So we've already kind of covered a lot of the conceptual stuff behind this in a lecture last year on DNA damage, and it might be worth looking at the podcast of that DNA damage lecture in tandem with this DNA replication lecture, because there are some parallels there between some of the mechanisms. One such concept is this idea of stoic hindrance, the idea that a lot of the enzymes which are involved in carrying out these processes, such as DNA replication, work in a sequence-independent manner. So DNA polymerase doesn't work and doesn't make the right decisions based on the sequence of the DNA that's being replicated, but rather the shape of the DNA molecules which it encounters. And why is that important? I'm being a bit cryptic. So the reason it's important is that's one of the ways in which DNA polymerase ensures that when it's replicating DNA, it adds deoxytrinucleotide phosphates to the growing DNA strand and not the far more prevalent ribonucleosides triphosphates which exist in the cell. So just for those who may have forgotten, a ribonucleoside triphosphate, that's the base that gets used up when you're transcribing a gene into mRNA. So the component parts of the mRNA molecules, the A's, the U's, the C's and the G's, they're ribonucleoside triphosphates. Very, very similar to dioxyribonucleosides deoxyribonucleoside triphosphates, which are the A's, T's, C's and G's, which are the constituent parts of DNA. Yeah. So the ribonucleosides, the RNA components, they're around in the cell at a much higher level than the DNA components, yet DNA polymerase never ever mistakes a ribonucleoside base for a deoxy ribonucleoside base. In other words, it never puts one of the RNA components into DNA when it's replicating the DNA genome. And how does it do that? How is that possible? Well, it's possible because of the fact that DNA polymerase is able to monitor very carefully the shape of the DNA double helix that enters the DNA polymerase active site. Yeah. So if, for example, here a ribonucleoside, an RNA base, was somehow found to be incorporated or attempt to be incorporated at this position, the shape generated in a double helix here will be very subtly different to the shape that you would expect to see were this a DNA component, a deoxyribonucleoside triphosphate. And that slight difference in shape is detected by this a quite complex DNA polymerase enzyme, which is responsible for synthesizing the DNA. So let me just explain a little bit more about the structure of this DNA polymerase enzyme and try to explain how that actually works. So obviously DNA polymerases are the enzymes which synthesize or polymerize new DNA. And atomic chemistry really, which has only been around for the last 20 years or so, maybe 25 years now, has revealed to us that the structure has enabled us to see the structure of DNA when bound to DNA polymerases. So in other words, you can look at the structure of a DNA polymerase when it's actually bound to a double-stranded DNA molecule or a DNA molecule that's been synthesized. And what people have noted is that the structure of the polymerase is anecdotally similar to the structure of a partially closed right hand. Yeah. So you have three main domains within the DNA polymerase enzyme. It's often easier to try and get your head around this if you think about these different domains as they're depicted here. So you have a palm region, the finger region of the polymerase, and the thumb region of the polymerase. So I've depicted that here. This isn't my hand, although it could be on a Friday night. You've got a thumb region here whose job it is primarily to grip the DNA substrate, as exemplified by this rather poor looking pint of lager. The thumb makes sure that the DNA polymerase keeps a tight grip on the actual DNA molecule and makes sure that the DNA molecule keeps near the active site of the enzyme during DNA synthesis. The palm region here, which is gripping the vast majority of the glass, that's the bit that's responsible for the actual nuts and bolts, the actual synthesis of DNA, the addition of a new base to the growing DNA molecule. And the finger region here, well, that's the bit that kind of regulates and controls the contact between the enzyme and the DNA molecule itself. So let's get rid of the pint and show this in a little bit more scientific way, if you like. So what we have here is a DNA molecule that's been synthesized. We've got the growing strand here and the template strand here. So this strand is a strand that's acting as a template. These are the bases that have been added to the growing strand of the DNA molecule. And what the DNA polymerase does is it kind of brings each of these nucleotide template positions into the palm region of the polymerase enzyme, which is the active site of the enzyme itself. And there's actually two metal cations, which are also present in the palm region of this enzyme, which are essential for the important processes of actually adding the DNTP, the nucleotide base, to the growing DNA chain. So you need to go away and do a little bit extra reading about this in the core textbooks. It's a very good chapter on this in Molecular Biology of the G, which explains this in a lot more detail than I've got time to go into today. But effectively, it's the palm region of these metal cations which are responsible for the actual addition of the nucleotide to the growing DNA strand. The finger region of the polymerase, well, that's responsible for kind of bending and manipulating the incoming nucleotide in such a way that it makes contact with A, the DNA template, and B, these metal cations which are required for the fusion of that nucleotide onto the growing strand of the DNA molecule. And the thumb region, well, that's responsible for just keeping a tight grip on the entire thing. And keeping a grip on the entire thing affords something called processivity to these enzymes, which we're going to talk more about later on, but in effect reflects the fact that each time DNA polymerase binds to the double-stranded DNA molecule, we don't just get the addition of one base, we get the addition of many bases. So for every binding of DNA polymerase to the DNA template here, it's able to add numerous, in some cases many thousands nucleotides to the template strand before it dissociates. Yeah, that's possible because of something called processivity. Processivity is possible because the thumb region of the polymerase enzyme affords a nice tight grip around the DNA molecule during the process of DNA synthesis. So, DNA replication itself then actually occurs in eukaryotic cells at a structure that's termed the replication fork. So there's a reason why the cell opts to copy both pieces, or if you like, both strands of the DNA molecule at the same time. It could be argued that it makes sense for it to copy one strand first and then the other strand, based on the fact that we have this lagging strand issue, which we'll talk about in a second, and you may have encountered at level 1 in cell biology. But it doesn't do that. It wants to copy both strands at the same time. The main reason it wants to do that is it's not a good idea to have single-stranded DNA lying around in the cell for any period of time. The reason it's not a good idea is if a break occurs in that single-stranded DNA, it's much more tricky for the cell to repair that break than if a break occurs in one strand of DNA when it is already associated with its sister strand, with its complementary strand. Those instances, those single-strand breaks in a double-stranded DNA molecule, very easy to repair. Single-strand breaks in DNA that is already single-stranded, very difficult to repair. So for that reason, it makes sense to replicate both strands at the same time. However, a key fact of DNA synthesis is that DNA polymerase cannot synthesize DNA de novo, so that means from scratch. So DNA polymerase can't just bind to a template and then start copying the information in that template into a new strand of DNA. It just doesn't work like that. What you actually require is a structure called the primer template substrate structure. Yeah. So this is the same concept as what we have in PCR, which we already covered it last year. So in PCR, you need to design PCR primers. The PCR primers bind to, in the case of PCR, a specific region in the template, and those primers then allow the recruitment of DNA polymerase, Taq polymerase, to that primer template substrate, and DNA polymerase then goes about copying or synthesizing your chosen stretch of DNA. It's the same principle during DNA replication in the cell. The cell requires a primer template substrate structure to be formed. Yeah? So for that reason, the actual process of DNA replication doesn't actually begin with replication of DNA, but it begins with the synthesis of a short RNA primer. And this RNA primer is actually synthesized by a special multi-subunit DNA polymerase called DNA polymerase alpha, and which is also sometimes called the primase enzyme. Yeah. So what primase does then is it produces a short RNA primer which is complementary to the DNA in the template strand. And this RNA primer then binds to the complementary region in the DNA strand to form a primer template substrate to which DNA polymerases can then be recruited, and DNA polymerases can then go about the business of actually replicating and synthesizing DNA. Yeah? Now, we've already talked about during the chemistry of DNA replication the fact that DNA synthesis must occur in a 5' to 3' manner. Put quite simply, it cannot occur in any other way. Yeah? The 5' triphosphates attack the 3' OH group of the previous nucleotide. So it's always added in a 5' to 3' manner. So because of that, we have quite a big issue with regards to the replication of the lagging strand, or so-called lagging strand of DNA in the double-stranded DNA molecule. So obviously synthesis of the leading strand here is very straightforward because that occurs in a 5' to 3' manner. So here the DNA polymerase enzyme can bind to the primer template substrate and then just start moving along this template strand, incorporating the complementary bases in the newly synthesized strand here, and that process can, in theory, go on indefinitely. The same is not true on the other strand, on the lagging strand, because on the lagging strand, what you have is you have a situation whereby primase will produce a small RNA primer that will bind to the DNA template, but then because DNA synthesis occurs in a 5' to 3' manner, the DNA polymerases will copy the template information here in a 5' to 3' manner, but eventually we'll get to the end, if you like, of this linear chromosome here, this linear piece of DNA. And what then will be required is, as this double-stranded DNA molecule unzips, primase will need to add another RNA primer further back here. DNA polymerases will come in and copy the DNA information again, and you will generate another copied piece of DNA, and so on and so forth, until you have loads of these fragments decorating the lagging strand of the DNA template. And these DNA fragments are what are known as Okazaki fragments. And I hope that you've already covered a lot of that in the level 1 module, cell biology. So as DNA replication proceeds, you have a nice simple situation on the leading strand here, where DNA polymerase just copies, in effect, infinitum almost, to a certain extent at least, the information in the leading strand, but the lagging strand is copied by virtue of many Okazaki fragments which decorate its entire length. Now, as this process is proceeding, the cell encounters another issue, and that issue is down to the fact that you're unwinding the double helix of the DNA molecule. So to get access to each strand of the DNA molecule, you need to kind of unzip the DNA molecule, and you can think about the similar process as if you have two pieces of string which are twisted together, and a knot in one end of the piece of string, and then you get your finger and you start moving your finger down that piece of string. What will happen is you will start getting coils forming beneath where your finger is. Yeah. If I'd be more prepared, so I can't do this on the podcast. I may do it in the lecture if I'm prepared. I may forget to bring the string, but try it at home. Get two pieces of string, tie a knot in one end of the two pieces, twist the two pieces around so it looks like the double helix of DNA, and then move your finger down to separate those two pieces of string. You'll see beneath your finger you get these kind of supercoils forming. The same thing happens during DNA replication. If you don't resolve these supercoils, then you end up with a problem. And the enzyme which is responsible for undoing these supercoils is an enzyme called topoisomerase. So topoisomerase works downstream of the replication fork to get rid of these supercoiled regions. But what is it that actually unwinds the DNA in the first place? Well, the thing that unwinds DNA in the first place to allow access to the templates is this DNA helicase enzyme. So we'll talk more about DNA helicase in a short while. This is recruited along with a lot of other things to the replication fork, and it's DNA helicase which unwinds those two DNA strands to grant access to the information which is then going to be copied during DNA replication. Now, I've already said that single-stranded DNA is the sort of thing that the cell doesn't like to have around, because if it's damaged, it's difficult to repair. One of the ways in which you can stabilise a single-stranded DNA to try and prevent damage is to coat it with a protein, and that's actually what happens. Once helicase starts unwinding the double-stranded DNA, the single-stranded DNA is actually rapidly coated in a protein called RFA. So this is what it's called in humans. It's called single-stranded binding protein in other organisms, such as bacteria, etc. And it's the binding of these single-stranded binding proteins, or RFA, that stabilises the template strands, these single-stranded DNA template strands, prior to replication. So you've got three processes going on at the replication fork. You've got helicase, which is unwinding the double-stranded DNA to allow access to the complementary strands. You've got RFA, which is binding to these single-stranded DNA molecules to stabilise them prior to replication by DNA polymerase. And you've got topoisomerase working ahead of the replication fork in order to get rid of these supercoiled regions which will form as you unzip the double-stranded DNA molecule. If topoisomerase wasn't present, eventually these supercoils would get so bad that they would prevent the function of helicase and the progression of the DNA polymerase enzyme, and DNA replication will come to a halt. So the topoisomerase is doing a very important job as well. So DNA polymerase alpha or primase, as we talked about earlier on, This is a very important enzyme because it's this enzyme which synthesizes this short RNA primer, which is absolutely required to form this primer template substrate structure to which DNA polymerases, other DNA polymerases, can then be recruited. Yeah. So DNA polymerase alpha produces this RNA primer, and it in itself can start copying the DNA, synthesizing the new DNA. But it's very poor at doing this. It has very poor processivity. So in other words, it's very slow at copying the DNA. So therefore, it needs to be replaced once replication's underway. So once you've got the process of DNA replication going, once primase has made the RNA primer that's needed to form the primer template substrate structure, it's replaced by the cell. And it's replaced in humans with two polymerases, DNA polymerase delta, which replaces primase on the lagging strand, and DNA polymerase epsilon, which replaces primase on the leading strand. So once primase has fulfilled its role, if you like, it's got replication going, delta replaces it on the lagging strand, epsilon on the leading strand. Now the difference between these two DNA polymerases and DNA polymerase alpha is the fact that these polymerases have very high processivity. So they're very efficient and very quick at replicating DNA. So for example, these enzymes are capable of perhaps adding around 10 to the 4 to 10 to the 6 nucleotides per binding reaction. Yeah, so that's about 1 million nucleotides can be added by DNA polymerase delta every time it binds to a primer template structure. Now, it's interesting that if you get rid of all the other proteins that are at the replication fork and just leave DNA polymerase delta there by itself, this figure drops to around 100 nucleotides. So in other words, there's something else in addition to DNA polymerase delta and DNA polymerase epsilon that's present at the replication fork that's largely responsible for the processivity, the efficiency and speed with which these DNA polymerases operate. So there's a YouTube video here that that won't work in this podcast. I can't play it, but you'll see it in the lecture, and obviously you can play it yourself alongside this podcast if you want to. The YouTube video effectively shows a car being pushed along a motorway by an articulated lorry. Looks a little bit scary, but it's there to emphasise how these processivity factors work. In other words, you have other proteins present at the replication fork which drive forward the polymerase and make sure that it's moving along the DNA template at a rapid speed and performing its function very efficiently. So those processivity factors are what are known as sliding clamps. And the one that's particularly important in humans during DNA replication is the protein pCNA. So pCNA is a very important protein. It's involved in a whole raft of different functions in addition to DNA replication. It's very important in DNA damage repair. But we're going to consider its role in DNA replication today, primarily. And it interacts with both DNA polymerase delta and DNA polymerase epsilon. So the sliding clamp itself, the protein, you can think of it as looking from the point of view of structure like a donut. And what happens is this donut structure can be opened and closed by a second protein which is called a sliding clamp loader. And in humans this protein is called RFC. And the RFC protein is responsible for the assembly of pCNA onto the actual DNA helix itself. And it does this in an ATP dependent manner. So this donut structure can be opened up like almost as you would imagine. At one point in the donut shape, there's a break which can be hinged open by RFC, and then this, if you like, donut shape clamps around the double-stranded DNA helix and then reforms this circle structure. Once you've got pCNA on the double-stranded DNA helix, it's then able to interact with the DNA polymerase enzymes. And once it's interacting with DNA polymerase, it's then able to process and move the DNA polymerase along the DNA strand at in a very efficient manner, at a very fast rate. We now know quite a lot about the actual physical structure of the pCNA protein. We know that within the circle here that wraps around the DNA molecule, there's just about enough room to allow several water molecules to actually be present. And current thinking is that these water molecules almost act like the kind of engine oil that you get in a piston and allow the sliding clamp to slide along the DNA molecule very rapidly and very efficiently and very smoothly. So it's a really ingenious system by which the cell's able to really drive these DNA replication enzymes along the template strand at a high rate of knots. Now, it's interesting that the same region of pCNA that interacts with the DNA polymerase enzymes is the same region of pcDNA that interacts with the loading complex RFC. So, is there a reason why these two different proteins, the polymerase and the RFC, the sliding clamp loading protein, interact with the same region of pCNA? Well, yes, there is, because obviously what you don't want to happen is for the polymerase for the sliding clamp to get loaded onto the DNA molecule in the absence of the DNA polymerase, and similarly, if pCNA is present on the double-stranded helix and interacting with DNA polymerase, in other words, if it's doing its job, if it's actually pushing DNA polymerase along, because DNA polymerase interacts with the part of pCNA which RFC also interacts with, there's no way that RFC can accidentally remove this processing factor, this sliding clamp, from the DNA halfway through the replication process, because the loading protein RFC simply can't get access to the binding domain it needs to remove the sliding clamp, because the polymerase enzyme is occupying that binding domain. Only when DNA synthesis is complete and DNA polymerase is released from the sliding clamp can RFC then come in and remove it from the DNA, the end of replication. So there's a couple of final issues faced by the cell. So we've talked about how DNA polymerases get recruited in. You have polymerase switching. Primase is replaced by the more efficient DNA polymerase delta and epsilon. These things are driven along by pCNA. So what happens when you've finished, if you like, replicating a chromosome, replicating the DNA? Well, one of the issues that you have when you finish replication on the lagging strand is that you're left with lots and lots and lots and lots of short RNA primers which have been synthesized and deposited by the primase enzyme in order to form these primer template substrate structures which are needed for the DNA polymerases to be recruited. Now, you can't just leave these RNA primers present in the DNA molecule, because RNA is a lot more unstable than DNA. They need to be removed and replaced by DNA nucleotides. So how does that happen? Well, a major component in that mechanism is this RNase molecule, RNase H. The H stands for hybrid, and it's called hybrid because this RNase molecule will recognize regions of double-stranded DNA where you have DNA paired with RNA molecules. And when it's recognized those regions, it will degrade the RNA from that section. Now, it's not as straightforward as that, because recent research has shown that there are a couple of other proteins also involved in this mechanism called FEN1 and DNA2. And these FEN1 and DNA2 proteins they're involved in the removal of this flap region which gets produced following digestion of these RNA nucleotides present in the RNA primer here. So RNase H will recognise this region, digest this RNA component here, and then FEN1 and DNA2 are responsible for snipping away the surrounding DNA prior to a DNA polymerase coming in and copying from this DNA template by virtue of this, if you like, primer region that exists here, which can now act as a primer template substrate structure. So perhaps I've not explained that. I'll just explain that again briefly, because I think I've rambled a little bit there. So this is the RNA primer that's left over. This is the primer that initially is placed there by primase. You need this so that the polymerase can come in and start copying this DNA strand here in a 5' to 3' manner. Yeah. So there'll be another one of these further upstream, if this is a lagging fragment, that's allowed the synthesis of this piece of DNA, this Okizaki fragment here. But you need to get rid of this RNA. That's done by RNase H, which will come in and digest it. The surrounding area is also cleaved away by two other enzymes, FEN1 and DNA2, and then the entire region's repaired by a DNA polymerase enzyme. The reason you can repair it with the DNA is that now we do have already a primer template substrate structure here by virtue of this replicated DNA region. So this can serve as a primer for DNA polymerase and allow DNA polymerase to come in and synthesize the complementary bases here, and then this final break which is left over here is joined by DNA ligase to generate an intact double-stranded DNA molecule. So what we've considered so far really is, if you like, what goes on with regards to DNA replication using one replication fork, and there's a quite complex but quite nice figure here which summarises the entire process. So this is the helicase enzyme. Remember, this is the enzyme that unzips the double-stranded DNA molecule. Once you start unzipping the DNA, very easy for polyepsilon to come in and start synthesizing the leading strand in a 5' to 3' direction, pushed along by pCNA. So that light pink here represents the initial RNA DNA primer that would have been left over by the primase enzyme prior to the primase being replaced by polyepsilon. On the lagging strand it's a little more complex. We have RNA primers that primase is putting down there. These serve as a primer template junction then for DNA pol delta to come in, which also is pushed along by pCNA. But rather than being able to just do a continual synthesis of the DNA, pol delta has to keep just doing short fragments of the DNA as and when the template becomes available as it's unzipped by the helicase enzyme. So this is what produces these Okazaki fragments, and then RNase H and FEN1 are involved in the removal of this RNA primer region here and replacing it with a DNA nucleotides. So that's what happens in single replication fork. But the truth of the matter is that DNA replication in your cells, it doesn't work like that. It works like that in bacteria, where you have a circular genome and there are one or two points within that genome where replication initiates, and then what you effectively have is a ripple around the entire circle of the DNA genome as the replication fork moves forward and copies the DNA sequence. But in eukaryotes, you don't just have one or two origins of replication, initiation sites of replication, you have lots and lots. So if you've got lots and lots of different origins of replication on a chromosome, how is all that regulated in DNA replication? How is it that each origin of replication only replicates once, for example, during S phase and not more than once? How is all that regulated? So it's regulated by something called the origin of replication complex. So I've just said that it's more difficult and more complex in eukaryotes. To exemplify that, rather than just having one or two origins of replication, each of your chromosomes actually contains many thousands of replicator sequences, as they're known in eukaryotes. And each of these can potentially serve as an origin of replication. So how does that work, and how is that regulated? Well, regulation begins when these replicator sequences are bound by an origin of replication complex. And this origin of replication complex will recognise a replicator sequence and bind to it. And then the first key event really that happens following binding of this ORC protein complex to a replicator is the recruitment of two other proteins, CDC6 and CDT1. Now the key thing about these proteins is they are only recruited during G1 phase of the cell cycle. It's impossible for them to be recruited at any time other than G1. Once these two proteins are recruited, they in turn then recruit a group of proteins called MCM2-7. It's also, or generally known just as MCM. And this MCM protein complex is actually a helicase, and it's the helicase which eventually ends up getting loaded on to the DNA at the replication fork. When CDC6 and CDT2 in G1 phase recruit MCM, you have then formed what is known as a pre-replicative complex, or pre-RC. Now the key thing to understand is that whilst this pre-RC complex is formed at replicator sites along the genome in G1, it does not fire until the cell has entered S phase. So what you've got is a loaded gun in effect. Once you assemble this helicase enzyme at the replicator site, you're kind of ready to go with regards to replicating the DNA, but the trigger isn't pulled until the cell enters S phase. Yeah? So why is it set up like this? Well, this sort of temporal separation of assembly and firing of replication facilitates regulation. It allows the cell to regulate. Yeah. So it's very important that when a cell regulates and undergoes DNA replication, that process is pretty rapid. Yeah. So you need to fire all of your pre-replication complexes more or less at the same time in order to get the vast amount of information contained within the eukaryotic genome, 4 billion base pairs, remember, of DNA, replicated in a timely fashion. So S phase in human cells doesn't take very long at all to complete. Yeah? To say that you're copying 4 billion base pairs, it takes an hour or two to complete. And it completes so quickly because many thousands of origins of replication site are all preloaded with pre-replication complexes and poised to fire, poised to initiate DNA replication once a signal's received and the cell enters S phase. We've already talked about what those signals are. The signals which are controlled by cycle-independent kinases within the cell cycle. So let's look in more detail at the exact signal which triggers firing of the pre-replication complex in S phase. Well, again, the regulatory proteins involved are protein kinases. Typo, apologies. In particular, CDK and DBF4. So these two protein kinases come along and in S phase they phosphorylate CDC6 and CDT1, which mark those two proteins for degradation by the proteasome. Once these two proteins have been degraded, numerous other factors are recruited to the replication, origin of replication, and what you have effectively is a burgeoning replication fork forming at this point in the genome, and DNA synthesis effectively begins. So this is shown here in this nice figure taken from molecular biology of the gene. This is your pre-replication complex. These are the two kinases we just talked about. They phosphorylate CDC6 and CDT1, which lead to those two proteins being degraded. Once they've been removed from this pre-replication complex, that then allows the recruitment of all other factors such as primase, DNA polymerase delta and epsilon to this replication fork. It also licenses the helicase MCM to start unwinding the DNA and for replication to begin. The second key thing about this process is that once you've pulled the trigger on the gun, once a pre-replication fork has fired in S phase, it cannot fire again until the completion of G1, until the daughter cells enter G1. The reason it can't fire again is because CDC6 and CDT1, which are required to form the pre-replication complex, are only found in the cell and only recruited to the pre-RC during G1 phase. So in other words, once you've undergone one round of replication from a replicator site, once this replication complex is fired, it's impossible for it to fire again, for the simple reason that it's impossible to recruit the correct proteins, in other words CDC6 and CDT1, until the G1 phase of the cell cycle, by which time the cell will have undergone mitosis, and rather than having one cell you'll have two, and the entire process of cell division is ready to occur all over again. So to re-emphasize, it's the absence of these protein kinases in G1 and therefore the presence throughout the rest of the cell cycle that ensure that each of the thousands of origins of replication only fire once per cell division. So these two kinases which are responsible for phosphorylating CDC6 and CDT1 aren't around in G1 phase. Because they're not around, these things aren't degraded. Because they're not degraded, they can be recruited to the pre-replication complex, but only in G1. At every other point in the cell cycle, these proteins get chewed up and get degraded, and you can't form these pre-replication complexes anymore. What it means is that complexes form in G1, fire in S phase, and then are unable to reform until the cells have gone right through cell division and divided, and enter G1 again, at which point CDC6 and CDT1 can be recruited to the pre-replication complex again, and the entire process can begin anew. So there's a final thing to consider regards to replication, and that's the so-called end replication complex. So this is a problem because, again, of the virtue of this 5' to 3' directionality to DNA synthesis, and the fact that you, on the lagging strand, have to synthesize DNA by virtue of these Okazaki fragments. Now, if you're always requiring an RNA primer to be placed down in order to produce an Okazaki fragment, eventually you're going to get to a point where you can't copy the very end of the DNA sequence because there's no DNA sequence here for which an RNA primer can anneal to allow synthesis of the complementary region. Yeah, it's impossible to copy this last bit of the 3' end of the DNA chromosome. So how is this problem resolved? It needs to be resolved. If it wasn't resolved, the result would be that one of the 4 replicated chromosome sisters during each S phase would be shorter than it's supposed to be. It would be shorter in length. And what that would mean is that over subsequent generations, you would get a continual shortening of the chromosome ends until such a point that the chromosomes start to become unstable, because this shortening will have started eating so far into the chromosomes it might have started to actually delete genes and other important regulatory regions of the DNA. So it's very important that the cell solves this problem. How does it do it? Well, the end replication problem is basically overcome via an enzyme called telomerase, which is a very clever, complex enzyme. And it overcomes this in conjunction with these specialised telomeric repeat regions of DNA which exist at the end of eukaryotic chromosomes. So when you get to the end of a chromosome, what you actually see is that the DNA sequence is comprised of many repeats of this sequence in humans, TTA, GGG. The second thing that you see is that actually the 3' end of the chromosome extends beyond the 5' end as single-stranded DNA. So you don't just get a flat-ended, blunt-ended, double-stranded DNA molecule at the end of the chromosome. The 3' end of the chromosome sticks out and continues this telomeric sequence, this TTA, GGG run of nucleotides. And it's to this 3' overhang that the telomerase enzyme can bind. And it can bind to this 3' overhang because a telomerase enzyme is actually what's known as a ribonucleoprotein. Yeah, so it's part protein, part RNA. And the RNA component of telomerase so happens to have complete complementarity to this repeat telomeric sequence that's present at the end of chromosomes and in this 3' overhang. So what that means is that telomerase can recognise these repeat sequences that are present in this 3' overhang, and the RNA within telomerase can bind to those complementary base pairs in such a way that the telomerase enzyme is recruited to the 3' end of the chromosome. Now the final thing about telomerase that's clever is the fact that it possesses something called reverse transcriptase capability. So reverse transcriptase is the ability to, rather than synthesize mRNA or RNA from DNA, it's the reverse of that. It's the ability to synthesize DNA from RNA. And what it actually does is, using a DNA RNA primer template, which is still required in order to synthesize new DNA, it uses this reverse transcriptase activity to copy DNA here in the 3' end using its own RNA as a template, thus extending this 3' overhang and preventing the degradation of the end of the chromosome. So that's summarised again in this figure here. So what I've just told you, this is the 3' overhang, this is the repeat region of the telomeric region, this is the RNA that's found within telomerase, and you can see it binds complementary to the telomeric repeat here, allowing telomerase to be positioned correctly. You then get elongation of the telomere region of the chromosome via the reverse transcriptase functionality of the telomerase protein. So in other words, you start adding DNA nucleotides to this 3' overhang of the chromosome using the RNA sequence as a template. And then the telomerase enzyme itself, once you've added these complementary sequences, can slide along and continue to add more telomeric sequence until such a time that the 3' overhang and the end of the chromosome is sufficiently long so that the chromosome remains stable. So how is the entire process regulated then? What stops telomerase from just indefinitely extending chromosome length? Well, one of the things that's important for regulating the length of telomeres are specific proteins which bind to the telomeric region itself. So these come in 2 flavours. There are proteins which bind to the double-stranded region of the telomere. So these are the regions of the telomeric repeats which are found near the ends of chromosomes. And also single-stranded binding proteins such as POT1, which will bind to the telomeric repeat that's present in this 3' overhang region, this region here of the telomere. And it's thought that the presence of both these types of proteins actually functions to inhibit telomerase activity. And it does so in kind of a sliding scale. And what I mean by that is that if you've got very few of these proteins present, in other words, if the telomeric repeat is very short at the end of the chromosome, then telomerase activity is very high because there are too few of these proteins to inhibit its function. When telomerase activity is high, it will add lots and lots and lots of these telomeric repeats to the end of the chromosome, thus extending chromosome length and making sure the chromosome end is stable. Conversely, if the chromosome end, the telomere, is very long, there will be lots of these binding proteins present, their inhibitory function will be very high, and telomerase will know, well, telomerase will be inactive, and the cell, if you like, if you're thinking of it as a sentient being, which it isn't obviously, will know that the telomere length is sufficient to confer stability to the chromosome, therefore telomerase activity isn't required. But what about the 3' overhang itself then? We've already talked about the fact that there's, last year and just in this lecture, that the DNA damage monitoring mechanisms don't like single-stranded DNA, but effectively you've got a single-stranded piece of DNA at the end of every chromosome. How is that dealt with? Well, it's dealt with, people think, by the chromosome forming what are called these so-called T-loop structures. So this is where the 3' end of overhang actually loops backwards onto itself and forms a loop at the end of the chromosome structure. And it does this by incorporating itself into the double helix region of the end of the chromosome, the telomere, by virtue of base complementarity pairing between the 3' overhang and the telomere repeats that exist at the end of the double-stranded chromosome. Excuse me. And it's thought that these T-loops structures effectively make the end of chromosomes look safe to the DNA damage monitoring mechanisms. They don't spot the fact that you've got a DNA break at the end of the chromosome or that you've got a single-stranded piece of DNA. And it's believed, recent research has shown that these two proteins, TRF1 and TRF2, are involved in the formation of these T-loop structures. So to finish with then, a few examples of what happens when enzymes involved in the process of DNA replication go wrong. Well, there's a very rare syndrome called Werner's syndrome that arises from mutations in the WRN gene. WRN is actually a DNA helicase that's known to interact with that DNA polymerase delta, so it's involved in the unwinding of the double-stranded DNA template during DNA replication. And when you get mutations in this, interestingly, the symptoms include cancer, which you'd associate with obviously errors in DNA replication, but also accelerated aging and associated illnesses with accelerated aging, such as diabetes and atherosclerosis, etc. So it's also interesting, therefore, that WRN has been shown to be involved in the maintenance of telomere length. So it's also a helicase which functions during telomere length, maintenance of telomere length, which we'll talk about a little bit more towards the end when I just talk a little bit more about telomere function. And you'll see why the fact WRN is involved in monitoring telomere length might be important with regards to the symptoms which people display when you have mutations in this gene. This is obviously an image taken of the same girl from the previous slide, but at age 48 she appears much older, like a woman in her 80s, so this premature aging phenotype which is associated with this illness. Another disease which arises indirectly from DNA replication errors is Huntington's disease. This is an autosomal dominant disorder that arises from mutations in the IT15 gene, and it's a lethal disorder which some of you will learn a lot more about because it's one of the topics for the group presentations. There's no known cure or treatment for Huntington's disease at the moment. It arises because of something called DNA slippage. So this occurs during DNA replication when the DNA polymerases encounter a run of these trinucleotide repeat sequences, these C, A's and G's. Now this particular type of trinucleotide repeat, when you have many of these stacked together, so a run of say 20, 30, 40 of these things stacked together, it interferes with the ability of the polymerase enzyme to move along the DNA strand and copy efficiently. And what that can lead to is a situation where one of the polymerases runs away from the other. Now if that happens, you can effectively rip apart the replication fork, which is a catastrophic event, and the cell wants to make sure that doesn't happen. So to make sure that doesn't happen, what can occur with these trinucleotide repeat regions is a looping of the template DNA. And what this looping achieves is it allows this strand which is falling behind because of the presence of these trinucleotide repeats to catch up with the other strand and prevents damage to the replication fork. Unfortunately, it can also result in the insertion or deletion of these trinucleotide repeat sequences in one of the replicating chromosomes. If you get insertion of these CAG repeats, then it can lead to Huntington's disease and other illnesses such as Fragile X, which occur when you expand these trinucleotide repeat sequences within our genes. So just to finish with them, because it's quite interesting, telomerase is also very important in human disease. We know that telomerase length correlates with the age of a cell, for example. So if you look at cells which are grown in culture, contrary to what we thought initially, which was that if you took cells out of a body and grew them artificially in a lab, they'd just divide indefinitely. We know that that's not the case. Cells have a Hayflick number associated with them. Hayflick related to the guy who realised this fact with regards to cells having a limited lifespan, if you like. The fact that they'll only go through a certain number of divisions before, if you like, stopping dividing and effectively dying, if you like. And the fact that the thing that seems to regulate how many divisions a cell can undergo is the length of its telomeres. So as cells continually divide in culture, telomere length shortens until such a point that it's so short that the DNA chromosomes become unstable. Chromosomal destabilization leads to massive genetic damage and cell death eventually. There are similar parallels, people believe, in the aging of organisms, therefore. So it's been thought for a long time that telomere length might relate to actually the aging of an organism, the aging of humans. And one possible way, therefore, of preventing aging or slowing aging would be to increase the activity of telomerase. Now, this guy is Juan de Pons, who was a Spanish explorer who famously looked for the so-called fountain of youth in the New World when during the invasion of those lands by Cortez, etc., during the 17th century. So that's where the de Pons effect comes from, which has been applied to telomerase in certain research articles. Unfortunately, as it says here, increasing telomerase activity is a double-edged sword. What happens if you increase telomerase activity, it turns out, is that you transform the cells, so the cells become cancerous. In other words, they start dividing uncontrollably, genetic instability occurs, and eventually cancer is a result. So that's a bit of a downer, it's a bit of a dull moment with regards to us all living forever. But we shouldn't give up hope yet, because there is recent research that shows that, at least in theory, manipulating telomerase activity may be able to actually reverse aging, never mind slow it down. So there's a paper in Nature a year or two ago which did some work on mice. And what they did is they deleted the telomerase gene from mice, and what they saw is that when you deleted telomerase from mice, the mice underwent very rapid aging and developed a whole host of other illnesses associated with aging, such as osteoporosis and diabetes and neurological problems, etc., including brain atrophy, so shrinkage of the brain and death of neuronal tissue. This is exemplified by this figure here. So this is this is a mouse that's just been born, if you like, or at a young age. This is its brain, and this is its brain has been weighed here in milligrams. This is the same mouse that lacks telomerase expression 4 weeks later, and as you can see, 4 months later. And in this mouse, the brain has shrunk significantly. There's significant atrophy going on of the neurons. So this is a mouse that's undergone a premature aging, etc. Now, if you take the same type of mice, the mice which are unable to express telomerase, and in this mouse you switch back on telomerase activity, what you see is the brains actually reverse. This brain atrophy process is reversed, and the brains of these mice actually regenerate and start to redevelop. Not just the brains, the osteoporosis, the other aging-related diseases which the mice were suffering from are all effectively cured, and those tissues regenerate and become healthy again. So this caused a bit of a stir when it was published in Nature a couple of years ago, and points to the fact that telomerase certainly is very important with regards to the cell's control of aging. What the current thinking is that whilst it's obviously very important in the control of aging of organisms and cells, There are many other mechanisms as well which control aging of humans, and it's not a simple enough process that all you need to do is to increase the level of telomerase to slow aging or reverse it. If you do that, it has knock-on effects such as cancer, and there are other factors at play that we don't fully understand yet, which also might need to be manipulated before that goal can be achieved. But it really shows what, sort of things are in the pipeline, I think, with regards to molecular biology, and how, understanding the components of core cellular processes such as DNA replication can really have quite far-reaching ethical and scientific implications for future generations and for future scientists such as yourself. So just to summarise then, I won't read through this, but this summarises what we've covered in the lecture. There are additional links on blackboards for additional reading. You should definitely read the chapter in the Molecular Biology of the Gene textbook on DNA replication. This lecture is heavily based on that chapter. It does a far better job and a more complete job than I can do in the time afforded to us of explaining this process. So I really strongly urge that you read through that chapter, because if you do, you'll get a much better understanding of the process of DNA replication. So as usual, this has overran the lecture allotted time of 50 minutes. One of the advantages of being at a podcast. I hope it's been useful. By the nature of these podcasts, sometimes they're a little bit. I don't like to say ranty. I don't like to think of myself as a ranting lecturer, but they can tend to run off course a little bit. But hopefully, they serve the purpose. It remains a useful tool for you guys to listen over this material again during revision. Okay, so there'll be more lectures coming soon on medical genetics in this series. Thank you.